Hello, everybody. Welcome back to another biology class. I'm so glad you're here. Today, we're going to be looking at gene regulation and mutation. Big topics, so let's get into it. The guiding question for today is how do prokaryotes and eukaryotes regulate their genes? It's a good question. It's not too bad. New vocabulary include gene regulation, operon, mutation, and mutagen. Fun topics. So let's get into it. Review vocabulary would be prokaryote today, and hopefully you remember that a prokaryote is a single-celled organism that usually has a single loop of DNA and a very simplistic internal structure. No membrane-bound organelles like mitochondria or chloroplasts. You might find some ribosomes, but most of the enzymatic activity occurs in the membrane of the prokaryote, and in terms of size compared to a eukaryote, which is the type of cells that you have, it is very, very small. Now, gene regulation is the process of turning on or turning off genes. And it makes sense that cells of any kind would have some form of gene regulation because always producing a protein is very costly in terms of energy. And not being able to produce a protein is not useful because you have a wasted program that's not being expressed. And so you need to have a method by which things can be turned on. And these methods are generally dubbed operons, okay? And so you have an operator, which is an on-off switch, and it's one part of the operator, operon. And then you have a promoter where the RNA polymerase attaches and produces your protein, your mRNA transcript. Then you have a regulatory gene sometimes, which is maybe a master control gene that tells other genes to turn on and off. And then finally, genes that can have coding for proteins via translation, which is something we talked about in the last class. So the trip or tryptophan operon. So tryptophan synthesis occurs in five steps controlled by the trip operon. The trip operon is repressible, meaning that can be turned off. So in the presence of the tryptophan product, it turns it off. And so sometimes you might think of the tryptophan operon as an always-on operon unless turned off. And what this does is it means that the more tryptophan is produced, the more end product is produced, the more this operon is deactivated. And so that allows for a pretty constant production of protein at a moderate concentration, as opposed to a full-on as much protein as it can possibly produce all the time, right? So this allows for a level of protein control that is sensitive to the amount of product, which is great because if you have too much product, too much tryptophan in this case, it would actually get in between other molecules and inhibit, slow down, other chemical processes that might be necessary in the cell. Now, the lac operon is a operon that produces the enzyme lactase as the end product. And this operon is off by default unless a certain sugar called lactose, which is present in milk, is present in the environment. And what happens is the lactose sugar binds to the inhibitor for the lac operon. The inhibitor is removed, then things can bind to the promoter region and your RNA transcript, your mRNA can be produced. And then that mRNA can be translated into a protein, the lactase enzyme. ACE is usually the idea of something being split or enzymatically functional. So the lactase enzyme is the end product. And so these are known as operon systems, and these are primarily in prokaryotes, but also used in some genes in eukaryotes as well. They're probably the simplest and best understood forms of gene regulation because they were some of the earliest discovered. Now, controlling transcription. This happens at a bunch of different levels, and in eukaryotes can be particularly complex because there are many different things that will alter how much of something is transcribed. Okay, and so you have pre-transcription controls, things like whether or not your DNA is wrapped around a histone, right? That physically inhibits the ability of mRNA to be produced. Whether or not there's an operon system involved or a controlling system involved, an inhibitor that has to be removed because of some condition, that's also a pre-transcription level of control. Now, you have post-transcription levels of control as well, because after your mRNA is produced, those introns and exons can be employed to lengthen perhaps the poly-A tail, which will say how many times a protein is produced from a single mRNA. And then you have your post-translation control, which is the degradation of the mRNA over time as it's being transcribed. And so that's determined, again, by how many A's were added during post-transcription editing and that sort of thing. Now, the book jumps here to what is known as the Hox gene, because in eukaryotes, there are master control genes. Okay, you can think of this as a kernel program, right, or an operating system where you have your master control program that will communicate with subordinate programs that it talks to, and turns them off and on. Okay, and so the Hox gene is the gene that is responsible for the development of the body plan in insects and in some vertebrates. Okay, and what's interesting about the Hox gene and why books care about this is that small changes to the Hox gene can wind up resulting in large changes to the organism as a whole. And so it's thought that mutations in these types of master control genes, the PAX gene for the eye is another such master control gene. It's thought that mutations in these genes can drive the evolutionary process. Well, there are some difficulties with that idea because, again, mutations are almost universally harmful, and if you fiddle with these too much, you're going to get some negative results. And the origin of these genes to begin with is not well understood in terms of an evolutionary paradigm where you have to build it one piece at a time. Because a control gene like the Hox gene has a on-off mechanism, right? So some kind of mechanism by which it can be activated or inactivated. Otherwise, there would be no point in it. It has to have something that it produces that touches or turns on or turns off, conversely, other genes. So whatever it produces, the master control gene, has to be able to interact with subordinate genes or connected genes. Okay, and that is a multi-component system, right? You have to have your timing control, you have to have your production that can actually control other genes, and then those other genes need to actually do something. And before you have all of those pieces in place, you can't select for a better version of the control gene. And so there are some questions in terms of natural selection and mutation in terms of whether or not they can write this type of integrated systems level programming. But the book assumes that evolution can, so we will as well. Yeah, Hox gene. Body plans. Okay. And again, the Hox gene, the idea is that you can have slight modifications of the master control gene leading to large physical changes in the organism. Okay, RNA interference is another method of gene regulation, and this can stop the mRNA from transcribing its message, right? This interferes with the mRNA. So single-stranded, small interfering RNAs, another fun RNA type, and protein complexes bind to your mRNA, and stop transcription. Okay, so these can be produced from the nucleus and sent out to stop the transcription message after it's happened. And again, this is in response to some kind of external stimulus. Some kind of chemical signal is going into the nucleus, telling the interfering RNA to be produced. This interfering RNA then interacts with the target messenger RNA and turns it off. And so this allows for a really fine level of control of product concentration. Now, mutation is the idea that you have something change. Okay, that would be the simplest kind of definition. And in your cells, a mutation can occur in your DNA, your RNA, or your amino acid. Mutations that tend to persist occur in your DNA. And it can be a variety of different types of mutations. You can have a point mutation where maybe an A becomes a C because it was incorrectly copied. You can have frame shift mutations where your DNA, when it's it's damaged for some reason, and a big chunk of it is just reattached at the wrong point, and so you have a distance that the code is still intact, but it was moved over by maybe 7 nucleotides, for example. Okay, that would be a frame shift mutation. You can have deletions where just chunks of DNA are removed. They're deleted. They're gone forever. Maybe the end of your chromosome broke off and it has been lost to the nether, digested, and is gone for good. Now, the interesting thing to remember about mutations, again, 1, they're almost always harmful, and 2, for them to have any evolutionary significance, they have to occur in your gametes, right? Your eggs or spermatozoa, because those contain the information, the program that is passed on to your offspring. Anything that occurs in a somatic cell, that's all the rest of the cells of your body that aren't egg or sperm, any of those kinds of mutations, they could be beneficial, they could be harmful, but largely they're irrelevant in terms of evolution because they can't be passed on to your children. And so mutations, again, tend to be harmful, and that's where cancer comes from. Now, protein folding instability is what we care about when it comes to mutations, because mutations in your program of DNA result in a different folding or an unstable protein. So how your protein folds, and there's 4 levels of protein folding: primary, secondary, tertiary, and quaternary. If we haven't talked about that, maybe we won't. This is a freshman level course, so that's not super important. But there's 4 levels of protein folding. The first level is just what amino acids are put where, and that's determined by your DNA code. And so mutations in your DNA code alter which amino acids are put in which order, right? And that leads to your secondary folding, which is largely done because of hydrophobic or hydrophilic ends or R groups on your amino acids, little sections on your amino acids that will face inward if they're hydrophobic or face outward if they're hydrophilic. Okay, and then you have tertiary folding, which has to do with hydrogen bonding and disulfide bonding, which leads to stability. Okay, and any of these levels of folding can be affected. A quaternary structure is when multiple proteins come together. I'll just mention that briefly. But that is affected, but it has to do with whether or not the proteins will fit together properly. And that has to do with whether or not they fold correctly. Now, causes of mutations are generally dubbed mutagens, and they range from environmental factors to just accidental copy errors. Anytime your DNA is copied, especially in a eukaryotic cell, there are millions of bases, and so they have to be copied, and occasionally you're going to get a copy error. This would happen in a computer where you're copying a hard drive. If you copy a hard drive 1,000 times, eventually you're going to have an accumulation of errors, and whatever was on that hard drive is going to have problems. Okay, same thing's true with DNA. And so that would be considered kind of a natural issue. Now, mutanogens are external things that can damage your DNA. Cigarette smoke has carcinogens, chemicals, that are mutagenic. They cause mutations in DNA, which is why if you smoke too much, you get lung cancer. Alcohols are carcinogenic, which is why if you drink too much, you get liver cancer or maybe kidney cancer. Okay, types of radiation. Athletes who get a lot of X-rays will oftentimes get various types of cancers because X-rays are mutagenic. They will cause errors in your DNA. So anything that causes an error in your DNA is a mutanogen. Okay, radiation tends to be one of it. Ultraviolet radiation, being out in the sun too long, is a cause of skin cancer, which is the result of accumulations of mutations. And again, body cells, somatic cells versus sex cell mutations. Body cell mutations, they could be advantageous. They could be great and make you healthier, but they die with you. Sex cell mutations could be great and advantageous and passed on to your children, or they could be horrible and awful and passed on to your children. Okay, things like prostate cancer for males can be passed on to male children, and that's the result of a mutation in a sperm cell, okay, or conversely a woman's egg cell, right? Or breast cancer is the result of a mutation in some cases that is passed on through sex cells. Okay, so mutations, again, they're oftentimes harmful, and if they happen in your sex cells, your oocytes, eggs, or spermatocytes, sperm, can be passed on to your children. So, how do prokaryotes and eukaryotes regulate their genes? Well, the easiest answer is for both is operon systems. And then you have multiple levels in eukaryotes, like pre-transcription editing, post-transcription regulation, and post-translation regulation through things like RNA interference. All right, that was a whole lot, and we'll have a lot more in class. Thank you so much for listening, and I hope you have a fantastic day.